Anyway, yeah, I'm sorry, I think we're going to have some discussion during the coffee break, which will need to be fairly short anyway. Yeah. Well, which is the reminder that right now we should be on coffee break, but let's ignore that. Okay, I'm going to talk a little bit about the topic of pharmacogenetics. Quickly, the point is, can we use pharmacogenetics to personalize aspirin chemoprevention? Talk a little about the beginnings, and I think I'll skip the overview. So I think we've heard it over and over. We know now aspirin is potent in cancer chemoprevention. However, we know there are GI symptoms, including in some cases serious bleeding. And this is actually fairly frequent at a dose of 300 or 600. This is a common event, not to have the serious bleeding, but to have some upsets and some problems. At 100, it's actually not that high, and they seem to occur mostly at the beginning, as Nadir had said. Nevertheless, risk-benefit balance, we have already heard about what do we do healthy young individuals versus those with family history, predisposing inflammatory or high-risk conditions, older, prior polyps, higher body mass index, etc. And pharmacogenetic tailoring now argues to try to do some genetic testing to figure this out. So we would have, for example, on our risk-benefit balance here, people who have few risk factors, a high genetic risk for bleeding or side effects, versus people who have high polyp and cancer and cardiovascular disease risk with genotypes that would favor the efficacy of the drug, and their aspirin use may be more indicated than on the other side. So being able to advise on aspirin based on the genetic predisposition, and with that I mean the genetic predisposition that tells us about whether or not people will benefit and whether or not they will have side effects, results in risk minimization, reduced concern among both the patients and the clinicians, reduced liability, and hopefully a greater likelihood of response. Now, we have studied this topic already. I started this in the US already about 15 years ago, and to think about first what are target pathways that we want to look at in terms of efficacy and toxicity, but also think about metabolism and excretion, and here are some of the key genes for that. But I'm also going to mention briefly what's happening now on the genome-wide association scale, and all of this together has also been supported by pharmacokinetic studies, which we did in some collaborations. So the studies that, here's the pathway in a little bit of a greater form, include genetic variation in the pathways, functional effects, and of polymorphisms and NSAID use in risk and survival and pharmacogenetics. So this is the short of this pathway, and when you start thinking about where could we target genetics here for efficacy, of course COX-1 and COX-2 are the prime enzymes that we would want to think about, but there are many others. And first of all, we need to know, of course, is the inhibition by NSAIDs, but here's also something which is like prostaglandin dehydrogenase, the anti-tumor suppressor, the tumor suppressor gene, which is the genetic NSAID, in that it can degrade some of these prostaglandins. But up here are also pathways that are by themselves related to carcinogenesis and can influence the flux that goes down here, and it has to do with the lipoxygenases. And then we also know that there's regulation, especially of COX-2 through cytokines, growth factors, etc., and regulation through peroxides. So when we started out, we said, okay, these are all these different pieces. How can we genotype through this? And first, when we started, there was no dbSNP or other resources to use to really understand what genetic variation exists. And so we started out by sequencing the key genes here in yellow and just the exons to see what variation there is. And what highlights now in green indicates what we then captured in a pharmacogenetic study with a so-called tag-SNP approach, where you basically cast a broad net to understand the genetic variation in those genes. So what happened first by sequencing? Well, first of all, there are very few non-synonymous polymorphisms in here. That means those that change the amino acid sequence and the protein function, which means this whole pathway is conserved. There's evidence for selective pressure. We found some polymorphisms that are likely to affect function. And we also know that in particular COX-2, the key driver here, does not have any non-synonymous polymorphisms in Caucasians. And so we know this pathway is highly conserved, and the evolutionary pressure for this probably has to do with wound healing. This pathway is very important for wound healing, and you can imagine that this is what drives that there is relatively little variability. I'm going to skip the slide on genotyping strategies nearly exclusively. Just understand this has been building with various populations of polyps, colorectal cancer, colon cancer, rectal cancer. And we started out here by saying, here's our first net of 380 polymorphisms where we can look at the range of carcinogenesis. And then this has been followed by a number of other populations that we have studied, including the Women's Health Initiative, and as part of an interdisciplinary collaboration with biochemists, we did functional evaluation of some polymorphisms. This is what COX-1 looks like as our primary gene of interest. These are the non-synonymous polymorphisms. You see there are very few in the actual gene body. Here's one in any exon, and most of these are in the front here, which is actually more important for regulation. And this is the crystal structure. And, whoops. And just to highlight some of these initial findings, this indicates a gene-NSAID interaction from an epidemiologic standpoint. So this is a risk reduction of colorectal adenomas. These are people, in the front here, people who don't use NSAIDs regularly. The red ones are the regular users. And you see here we split up by a polymorphism in COX-1. And you see that the genotype plays a role among the non-users, whereas back here there's not much going on. This is actually a fairly small group. So this illustrates some evidence for gene-NSAID interaction as the proof of principle that we can do pharmacogenetics. And then we did publish, if you're interested, some on the functional effects of these polymorphisms. Now, I mentioned that metabolism is important here. In the liver are actually some of the most important genes, such as the UGTs, which involve glucuronidation of NSAIDs, including aspirin, but also many other drugs, or CYP2C9 as a major metabolizer. And here, just again, as a little teaser, some of the data that has just been published on one of the UGT genes, UGT-B15. And again, here the non-users are in the front, the current users are in the back, and you see that basically based on genotype you have a different pattern. Now, these were all preliminary initial studies. What really is now in the pipeline and unpublished just in poster form at this point is a meta-analysis of all these individual study populations where we try to say, if we look in 10,000 people, what are the robust signals that are left that help us to answer the question, who benefits from NSAIDs and who doesn't? And we used a two-stage approach in that we first cast a net in some populations and then attempted to replicate those. And this is an example of the interaction effects and their consistency for some polymorphisms and how consistent they are here. You see they're all on this side. So this is a significant gene-environment interaction that holds up and is statistically significant. And what does it actually translate to when we visualize this? So we see here the regular use of NSAIDs in the front in these populations, and you see that the efficacy, the benefit in risk reduction by more than 30% is limited to people who have a certain genotype in the 5-lipoxygenase activating protein. So much has been published. I tried to keep this short, realizing that some of my colleagues would have a lot to say. But these signals are now robust and can replicate. But what do we do about this? They come from the epidemiology. How can we take them to the clinic? So one big aspect is, of course, to do genotyping in clinical trials, and this is what Nadir has referred to as our wonderful collaboration, where we started out first by looking at the effects of the genetic polymorphisms on adenoma recurrence and toxicity in his clinical trial, the celecoxib trial. So this is a pilot study, but we have just received funding from the European Union to investigate this in a much larger scale in multiple trials. But one needs to keep in mind that there are not that many trials that have blood, and the results will never be in 20,000, unfortunately. So this is the pilot data from Nadir's PRECEPT trial. Just briefly, what happened is that people had to have had a colonoscopy of at least one adenoma of reasonable size or two smaller adenomas, and then adenoma recurrence was measured about 3.5 years after study initiation, and of course also symptoms and toxicity was assessed by the investigators for the on-treatment period and from the subjects afterwards. And we grouped these symptoms and toxicity in this admittedly small population into any cardiac, gastrointestinal, etc. And so this has been published last year in Cancer Prevention Research, and here are some of the findings that we would like to have. So we had some primary genes of interest that we studied, and here of greatest interest is cardiac symptoms and toxicity, because that was the factor that caused this trial to end. And just to give you an idea of what we can see, in the celecoxib arm here, there are some genotypes in prostaglandin E synthase that confer a vastly increased risk of cardiac toxicity. So if we had known this beforehand, those people should not have been on the trial, the people who have certain genotypes in PGES. And here's just to visualize what we also see with respect to genes and adenoma recurrence. You can see how there's a nice splitting of the curves depending on a variant in the COX-2 enzyme. Homozygous variant basically has a much higher probability of recurring, lower probability than the wild type, and the heterozygotes are in the middle. So I mentioned before that these were targeted approaches where we had candidate pathways based on the known biology. Now one can also say we can look under the lamppost, but often our prior knowledge is not that strong that we make the best decisions. And in collaboration with Andy Chen and other colleagues from the GECKO consortium, a study has been undertaken now to look for other signals by doing a genome-wide association study. So this is untargeted and empirical with 2.7 million SNPs that were looked at, and there was one region identified, in this case control design, that unfortunately I cannot talk about because it's not published yet. But overall one needs to keep in mind that there were very few signals besides this one, and if one tests for 2.5 million SNPs at the same time, one gets statistically punished very severely for doing so. And so by missing some things, these are probably false negatives. So if we have a pathway that we know about and it doesn't show up here, it may mean that just statistically you get punished for doing so many comparisons. So both approaches are complementary. So to summarize this briefly, pharmacogenetics of aspirin is really relevant with respect to the pathways of metabolism and excretion, for efficacy, side effects, and toxicity. It will help us to consider this by developing an indication for aspirin for cancer prevention, because we will be able to personalize prevention based on risk of bleeding and other side effects versus the benefit that one is likely to have. And there will be also potential for other considerations such as stratification by biomarkers or discovery of other pathways, for example with metabolomics, something that we are working on. So the goal should be hopefully to identify yet another blood test, and in this case one that helps patients and clinicians make informed decisions on whether to recommend ASS for colorectal cancer prevention, and this being particularly relevant for a higher risk profile. So what's the future besides this? I think this is a long-range forecast, but I want to say there are just a few other future steps. What I didn't talk about is the mutational status of the tumor, that we can also consider aspirin in the adjuvant therapy of cancer based on the data that have emerged and that Nadir briefly has mentioned. And there are clinical trials underway that this very inexpensive drug may help patients to be successful in reducing their risk of recurrence. And this may depend on mutational status. There's limited data at this point. There's been high publicity for PI3 kinase, but I think meta-analyses are needed because there's still some inconsistencies. These are initial data. So with that, I would like to thank my colleagues both at the Fred Hutchinson Cancer Research Center and, of course, at the NCT here in Heidelberg and my group and everybody else here for sticking around and lasting so long. Thank you.
Thank you very much for this very promising talk. Unfortunately, the time is far, and so as a clinician, for me, the most important message is that after these talks we heard in the afternoon, that aspirin does have an important role in the prevention of colorectal cancer, and it's up to us now to get this indication into our strategies, especially in our guidelines, to get a better help for the patients who do not want colonoscopy or who are at a certain risk, and so we have to change our strategies.
I think that perfectly sums it up.
Is there any important comment at this point?
Jürgen? No.
That's not the case, and I thank you all, and